So this is the second part of how to use a hemocytometer to count cells in a cell culture. So after you have successfully added cells onto your hemocytometer, as demonstrated in the previous video, you will simply place the hemocytometer onto the microscope stage like you would an ordinary slide, position one of the counting chambers over the light source, and then start with the 4x objective lens to get your initial focus. So I've already done that. So as you begin to focus, you should be able to find this square grid pattern. So this is the actual counting chamber itself, and as you can see, it is divided into a number of smaller and smaller squares. There are 9 large subsquares, and then each one of those is divided into smaller sets according to different dimensions. So these outer ones are subdivided into 4x4 grids. The center square, however, is divided into a 5x5 grid. And you're probably wondering, well, where are the cells? Well, depending on how good of a focus and how good you can see, there are a bunch of tiny little dark specks scattered all over the grid. Those are the cells. But they're so small we can't really count them at this magnification. So we want to go ahead and increase magnification, and that also allows us to use only a smaller portion of the hemocytometer. We're only going to be using the center grid, that is the 5x5 matrix. So make sure that that is centered in your field of view, and then go ahead and change up to the 10x objective lens, and then refocus. And there we are. All right, now again, you can begin to make out the individual cells a little bit better. But you're probably looking going, that is a lot of cells to have to count. The nice thing about our protocol is that we don't actually have to count all of the cells in this center square. In fact, we're going to only sample from our 4 corners. So you're going to go to the upper left, followed by the upper right, move down to the lower left, then to the lower right, and then finally back to the very center. Now, in order to get an accurate count, we actually want to count very closely at just those 5 sub-squares. So go ahead and reposition your view so that your upper left corner square is centered, and you're going to increase magnification one more time to the 40x objective lens. And as you refocus, you will see this square. Now you will know that you are on the correct square because you'll notice that the outer boundaries have 3 lines on them. And so that is the appropriate magnification. And so now we want to try to count up all of the cells that are present in this square, and we can still utilize the 4x4 grid inside this square to help us keep track of where we've counted and where we haven't. So we can first go to this upper square and start counting cells. And it looks like we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Looks like we got about 15 or so in that one. And then we move over to the next one and we count all of those cells. You will be given a hand tally counter, or basically a device that you click every time you count a cell so that you can keep track of your numbers and not lose count. So we would simply go through this cell, count up all of them, and then you would move across your field of view until you get to the opposite cell. And you can tell you've gotten to that edge because you'll get to a region where there are horizontal lines but no vertical lines. And so now I know I'm on that corner, and I'm going to go ahead and count those cells. Now, one kind of convention in counting cells with the hemocytometer is when you get to the cells that are on the very edges, and if you were to be counting cells that were actually adjacent to one another, say you were counting not just this very corner cell, but also the one immediately to the left of it. What would you do to count a cell that was right on the line between the two? And if you were first counting this one and then go counting the one on the left and then moved over to the cell on the right, would you double count that cell? And of course the answer should be no. But how do we make sure we don't double count? Well, the convention with hemocytometers is that when counting any particular square, any cells that are located on the left or the top border of a cell, if it's touching those border lines, those cells are counted in, but any cells that are touching the right or bottom boundaries are not counted, because then if you were to count an adjacent cell, those cells could be counted by other cells. So that's pretty much all you have to do. After you do the second cell, you go down until you find the corner cell, count all of those cells, and then scan across to the left, find the left lower corner. And once you've counted all those cells, then you're just going to go up 1, 2, up, and then 1 and 2 over, and get back to the center cell and count all of those. Once you've counted those 5 small cells, you are done with that counting chamber, and then you're ready to move on to another counting chamber. So that's the technique for using the hemocytometer. Now, after you get those cell counts, there are ways of calculating what the Fowler cell density is, and you will see those instructions in your laboratory manual.